Health Organization recognized the late Henrietta Lacks, a Black American woman whose cancer cells were taken without her consent or knowledge, and those cells helped revolutionize modern medicine. Here's a look by the numbers. In 1951, Henrietta Lacks sought treatment at Johns Hopkins Hospital for cervical cancer, where researchers collected her cells without permission. She died just 8 months after her cancer diagnosis at age 31, but her cancer cells somehow have lived on. Researchers discovered those cells double every 20 to 24 hours. They are seemingly immortal. While other cells scientists studied would die within days, hers are the first human cells ever cloned, and that has revolutionized biomedical research. These cells have since contributed to nearly 75,000 medical studies, helping advance HPV and polio vaccines, HIV/AIDS medications, in vitro fertilization, and they are still in use right now to research COVID-19. But this is not an entirely happy story. For 2 decades, Lacks cells were commercialized and distributed without her family's knowledge. But her story would gain national attention from the book The Immortal Life of Henrietta Lacks, and then the HBO movie with the same title. And her unjust treatment has been linked to other examples of medical racism, including the Tuskegee study that lasted from 1932 all the way to 1972. And today, the WHO Director General presented a posthumous award to her 87-year-old son, Lawrence, saying this is an opportunity to recognize women, particularly women of color, who have made incredible but often unseen contributions to medical science.
Imagine something small enough to float on a particle of dust that holds the keys to understanding cancer, virology, and genetics. Luckily for us, such a thing exists in the form of trillions upon trillions of human lab-grown cells called HeLa. Let's take a step back for a second. Scientists grow human cells in the lab to study how they function, understand how diseases develop, and test new treatments without endangering patients. To make sure that they can repeat these experiments over and over and compare the results with other scientists, they need huge populations of identical cells that can duplicate themselves faithfully for years. But until 1951, all human cell lines that researchers tried to grow had died after a few days. Then a Johns Hopkins scientist named George Guy received a sample of a strange-looking tumor. Dark purple, shiny, jelly-like. This sample was special. Some of its cells just kept dividing and dividing and dividing. When individual cells died, generations of copies took their place and thrived. The result was an endless source of identical cells that's still around today, the very first immortal human cell line. Guy labeled it HeLa after the patient with the unusual tumor, Henrietta Lacks. Born on a tobacco farm in Virginia, she lived in Baltimore with her husband and 5 children. She died of aggressive cervical cancer a few months after her tumor's cells were harvested, and she never knew about them. So what's so special about the cells from Henrietta Lacks that lets them survive when other cell lines die? The short answer is, we don't entirely know. Normal human cells have built-in control mechanisms. They can divide about 50 times before they self-destruct in a process called apoptosis. This prevents the propagation of genetic errors that creep in after repeated rounds of division. But cancer cells ignore these signals, dividing indefinitely and crowding out normal cells. Still, most cell lines eventually die off, especially outside the human body. Not HeLa, though, and that's the part we can't yet explain. Regardless, when Dr. Guy realized he had the first immortal line of human cells, he sent samples to labs all over the world. Soon the world's first cell production facility was churning out 6 trillion HeLa cells a week. And scientists put them to work, in an ethically problematic way, building careers and fortunes off of Henrietta's cells without her or her family's consent, or even knowledge until decades later. The polio epidemic was at its peak in the early 50s. HeLa cells, which easily took up and replicated the virus, allowed Jonas Salk to test his vaccine. They've been used to study diseases including measles, mumps, HIV, and Ebola. We know that human cells have 46 chromosomes because a scientist working with HeLa discovered a chemical that makes chromosomes visible. HeLa cells themselves actually have around 80 highly mutated chromosomes. HeLa cells were the first to be cloned. They've traveled to outer space. Telomerase, an enzyme that helps cancer cells evade destruction by repairing their DNA, was discovered first in HeLa cells. In an interesting turn of fate, thanks to HeLa, we know that cervical cancer can be caused by a virus called HPV, and now there's a vaccine. HeLa-fueled discoveries have filled thousands of scientific papers, and that number is probably even higher than anyone knows. HeLa cells are so resilient that they can travel on almost any surface. A lab worker's hand, a piece of dust, invading cultures of other cells and taking over like weeds. Countless cures, patents, and discoveries, all made thanks to Henrietta Lacks.